Faculty of Medicine, Kafrelsheikh University, Egypt. And she obtained master degree MD from Faculty Medicine, Cairo University, Egypt. She is a head of management and assessment unit, Faculty of Medicine, Kafrelsheikh University, 2020. And she works as postdoctoral researcher in Lab of Nano-Biotechnology research in biotechnology, cancer research, and cell biology at Okayama University, 2019. And she is member in Kafrelsheikh University unit. For harassment and violence against women, 2019. She works as member of Quality Assurance Units, Faculty of Medicine, Kafrelsheikh University, Egypt, 2017. And she was Assistant Lecturer of Department of Histology and Cell Biology, Faculty of Medicine, Cairo University, 2014. So I will stop sharing my screen to give you a chance. Dr. Amira will present now instead of.
Good morning. Good morning, Dr. Walid.
Then
Do you hear me?
Yes, I hear you. Amira, you are okay?
Dr. Walid, do you hear me?
Yes, I hear you, but your screen still.
Dr. Walid?
Yes, I hear you. Yes, it's playing now.
Okay.
I see your screen. So let's go.
Okay.
Thank you, Dr.
Okay. Could I start?
Yes, please.
First of all, I would like to thank dear Professor Dr. Ibrahim Tantawi for his kind invitation, and many thanks to Dr. Walid Saad for her great efforts to organize this symposium. And I would like to thank Professor Dr. Fukami for her support and all organizing and scientific committee members. Now, I will talk about COVID-19 and anosmia. The pandemic of coronavirus disease COVID-19 has caused a vast disaster throughout the world. There is increasing evidence that olfactory dysfunction can present in COVID-19 patients. Anosmia can occur alone or can be accompanied by other symptoms of COVID-19. However, the pathogenic mechanism of olfactory dysfunction and its clinical characteristics in patients with COVID-19 remains unclear. Multiple studies have demonstrated that the incidence rate of olfactory dysfunction in COVID-19 patients varies from 33.9 to 68% with female dominance. Post-viral anosmia is one of the leading causes of loss of sense of smell in adults, accounting for up to 40% cases of anosmia. What is anosmia? It's a partial or complete loss of sense of smell. This loss may be temporary or permanent. Common conditions that irritate the nose lining, such as allergies or a cold, can lead to temporary anosmia. More serious conditions that affect the brain or nerves, such as brain tumors or head trauma, can cause permanent loss of smell, swelling or blockage in the nose that prevent odor from getting to the top of nose. Anosmia is sometimes caused by a problem with the system that sends signals from the nose to the brain. What about COVID-19 anosmia? Patients with COVID-19 can present sudden onset of anosmia with other symptoms, without any other symptoms. Before the onset of anosmia, other mild symptoms such as dry cough or may also be presented. 54 out of 114 confirmed COVID-19 patients presented with anosmia. The study also found that the patients generally developed anosmia 4.4 days after the onset of the SARS coronavirus 2 infection, with a duration of 8.96 days. The studies showed that individuals with smell disorder tend to have a taste disorder, suggesting a probable association between the two. To date, study using self-reported questionnaire analyzed the prevalence of smell and or taste disorders in COVID-19 patients and influenza patients. It proved that the incidence rate of COVID-19 patients was significantly higher than that of influenza patients. Some studies reported that anosmia was the sole or primary symptoms uh, uh, of COVID patient confirmed by 6 odor smell tests. Another study performed a remote olfactory evaluation for 23 COVID-19 patients in home isolation, utilizing patient self-administered olfactory function test with decreasing concentration of denaturated ethyl alcohol solution. The data of the patient self-administered OFT can help with the early detection and isolation of COVID-19 patients. Based on research, olfactory dysfunction has a high incidence rate in COVID-19 patients in some European and American countries, while it rarely occurs in Chinese patients. It reviewed 5 articles about the clinical presentation of COVID-19 patients from China, comprising 1,556 cases with no reports of olfactory dysfunction. Researchers retrospectively analyzed the neurologic symptoms of 214 patients in Wuhan, China, and found that 5.1% of patients exhibited smell impairment. To our knowledge, this paper is the only study to date to describe olfactory dysfunction with COVID-19 patients in China. Analysis of electronic health record indicates that COVID-19 patients are 27 times more likely to have smell loss, but are only around 2.2 to 2.6 times more likely to have fever, cough, or respiratory difficulty compared to patients without COVID-19. Some studies have hinted that anosmia in COVID-19 differ from anosmia caused by other viral infections, including by other coronaviruses. For example, COVID-19 patients typically recover their sense of smell over the course of weeks, much faster than the months it can take to recover from anosmia caused by subset of viral infection known to directly damage olfactory sensory neurons. In addition, many viruses cause temporary loss of smell by triggering upper respiratory issues such as stuffy nose. Some COVID-19 patients, however, experience anosmia without any nasal obstruction and the absence of significant nasal inflammation or coryzal symptoms. The underlying mechanism for loss of smell in patients with COVID-19 have been unclear. I want to talk about the nasal epithelium. It is formed of 2 parts, olfactory epithelium and respiratory epithelium. Respiratory epithelium contains ciliated cell, brush cell, goblet cell, secretory cell, basal cell. Olfactory epithelium in the upper part of the nose consists of mainly olfactory sensory neuron and supporting cell like basal horizontal cell and microvillar cell, sustentacular cell, and goblet cell. It has recently been demonstrated that single-cell RNA sequencing analyzes that cells from the human upper air, including nasal respiratory epithelium, goblet, and ciliated cell, express high level of angiotensin converting enzyme 2 and TMPRSS2, suggesting that these respiratory epithelium cell types may serve as a viral reservoir during coronavirus 2 infection. Non-neuronal cells in olfactory epithelium and olfactory bulb, including supporting and stem and perivascular cell pericyte, express coronavirus 2 entry-associated transcripts and their associated proteins. This is a diagram about how coronavirus enter to respiratory olfactory epithelium by interaction between S protein and angiotensin converting enzyme 2. So SARS coronavirus 2 causal agent in COVID-19 don't affect olfaction direct by infecting olfactory sensory neuron and their targets in the olfactory bulb, but indirect by perturbing supporting cell bulk, supporting cell in olfactory mucosa. Bulk and single cell RNA sequencing revealed that supporting and stem cell in the human and mouse olfactory epithelium and vascular pericyte in the mouse olfactory pulp express ACE2, which is essential for coronavirus 2 cell entry. In contrast, ACE2 was not detected in either olfactory neurons or olfactory pulp neurons. Immune staining confirmed this result and revealed pervasive expression of ACE2 protein in dorsally located olfactory epithelium, sustentacular cells, and mouse olfactory bulb pericyte. This finding suggests that coronavirus 2 infection of non-neuronal cell types lead to olfactory dysfunction in patients with COVID-19. This is immune stain of human olfactory mucosal biopsy. Detection of ACE2 protein, take the green color, present in both sustentacular cell, which is like a receptor cell on the surface of olfactory mucosa, and the basal, horizontal basal cell only. And neurons of olfaction is not contain ACE2 protein. Anosmia seems like a Kirsch's phenomena, but it can be devastating for the small fraction of people in whom it's persistent. It can have a serious psychological consequence and could be a major public health problem if we have a growing population with permanent loss of smell. The researcher also hope the data can help pave inroads for question on disease progression, such as whether the nose acts as a reservoir for SARS coronavirus 2. Such efforts will require studies in facilities and that allow experiments with live coronaviruses and analysis of human autopsy data. More extensive studies, both basic and clinical, would need to be conducted to reveal this mysteries in the future. The similarity to human being, the SARS coronavirus 2 infection rhesus monkey can be used to study the pathological and physiological of olfactory system. Our group in Faculty of Medicine, Kafrelsheikh University, has a project about anosmia and take biopsies from the olfactory epithelium of COVID-19 patient for ultrastructure observation to better understand the pathology of olfactory dysfunction on COVID-19 patient and finds the unclear mechanism about transient and permanent anosmia and try to modulate anosmia treatment protocol. Thank you.
Thank you so much, Dr. Amira. Interesting lecture.
Wala?
Yes, I hear you. You are
If
hearing me?
any question?
You are hearing me? Okay. Yes, so interesting.
Yes, yes, I hear you.
Okay, it's a very important issue because it's a very rare sign and symptom to present in COVID-19 without any other symptoms, only anosmia, loss of smell, sudden acute loss of smell in patient with COVID-19 with no fever, with no cough, with no sneezing, runny nose, in viral infection like influenza, we can find running nose with loss of smell, but it's for 1 day, 2 day. In COVID-19, the issue will be more several days, up to 10 days loss of smell and acute sudden. It's very
More
annoying.
than this,
Yes,
some patient
no,
months.
yeah, if
And now new cases permanent anosmia.
yeah, you are right. When I read about anosmia at first of COVID-19 crisis, I found many papers, international papers, even in ENT societies abroad in America, point that there is a test to olfactory nerve for diagnosis of anosmia, and also there is a training to train COVID-19 patient to how regain so quickly the sense of smell again by smell more than 3 times per day, like rose, lemon, strong smells to can regain. So it's a very important issue. Thank you so much. I would like to accept any question for Dr. Amira. I see Dr. Sobhi have a question. So Dr. Sobhi, please. Dr. Sobhi, you hear me? Dr. Sobhi? Dr. Amira, may I ask you to stop sharing your screen?
Okay.
Thank you. Dr. Sobhi, حسب النبي, can you ask your question?
Hi, Dr.
Good morning, Dr. Sobhi.
Hi, Dr. Amira.
She is
Hi, doctor. I hear you.
Dr. Amira.
Yes.
Thank you for good lecture. I have a question. Acetylcholine series 2 as a receptor is found all over the cells or?
Dr. Sobhi, I can't hear you.
Have unstable internet. Your internet get some lag. So check your internet.
Now I hear you, Dr. Wala.
Yes, Dr. Sobhi have some problem in internet because I see it hang out. So I think there's some problem at Dr. Sobhi. So thank you so much, Dr. Amira, for your presentation. And I have one question till Dr. Sobhi can return connected. So you about your study in your faculty in Kafrelsheikh about loss of smell in COVID-19 patient, and you get some smears or some biopsy from epithelium. Do you suppose you any mechanism or any some sort of receptor damage or end nerve damage you find, or you didn't show any result with it?
Dr. Wala, we now search about permanent anosmia when it was transient. So treatment was just vitamins and something like this because it affects supporting cell only, and the olfactory neuron is not affected. But some cases reported is permanent anosmia, 3 months anosmia and 2 months anosmia. So we research about now what is the cause. Is this olfactory neuron is affected also or no? But till now we didn't find this answer.
Thank you so much. So I didn't receive any questions, so thank you, Dr. Amira. And
Thank you, Dr.
I hope when you finish your project, you show your result to our site. And
Very near.
yes, I am so interested to know why, because I am personally experienced this bad experience about loss of smell. So I know it's very annoying. Thank you, Dr. Amira, and I will I will see Dr. Our next speaker. So I will present him now. So our schedule will be some change. We will now I will welcome Dr. Bala to present his presentation till Dr. Amany sent her PowerPoint because still the problem is present. And I will present Dr. Amany PowerPoint from my side, sharing from screen, and she will comment it because it's to fix this problem. So Dr. Bala, please share your screen. You hear me, Dr. Bala?
Sorry, Dr. Wala. I have
trouble
troubleshooting my computer here.
Yes, Dr. Sobhi. Sorry, Dr. Bala, excuse me 1 minute. Dr. Sobhi, I see you have a question. You are hearing me, Dr. Sobhi? You had
Yes, I hear you.
Okay, you have question for Dr. Amira.
Yes. My question, Dr. Amira. Are you hear me?
Yes, doctor. You are welcome.
The receptor of acetylcholine stress 2 is present in all cells or specific tissue?
Specific cell on olfactory epithelium. Supporting cell and not olfactory neurons.
Only
only supporting. Only supporting cell. So, one cell called sustentacular cell and another one is basal horizontal cell and the pericyte.
This means COVID-19 is specific for olfactory only, but because there is no receptors in another tissue. But it is well known COVID-19 affects the respiratory system. So respiratory system or the cells of respiratory system should have acetylcholine stress 2. Because you know this receptors.
I speak about nose only, Professor, not all respiratory system.
Okay. This is my question. Thank you, Dr. Amira.
Thank you.
You are welcome, Professor.
Thank you, Dr. Sobhi. Thank you, Dr. Amira. So, Dr. Bala, please, I will introduce yours into your biography till you start the screen, share your screen. Okay. So, Dr. Bala Kanish, excuse me, is scientist at Laboratory Division, ICMR National Institute of Epidemiology, Chennai. She was awarded a doctorate in microbiology virology from University of Jadavpur, Kolkata. Previously worked at National Research Center of Disease Control, ICMR National Institute of Cholera and Enteric Diseases, Kolkata. And she has published 41 research articles, 2 book chapters, and 1 book. He got selected for Infectious Disease Fellow Program at American Society for Microbiology Conference, Microbe 2018 at Atlanta, USA, during June 7 to 11, 2018. He is a principal investigator for JSPS, Joint Research Project under the Prestige India-Japan Cooperative Science Program, and he is recently made an exchange visit to Professor Nobumichi, Professor and Head of Department of Hygiene, Sapporo Medical University School of Medicine, Japan, between 26 January to 11th February 2020. So welcome, Dr. Bala, and I will stop my screen to join your screen now. Then Dr. Hisham will present the next presentation. I'm so sorry about this disturbing in program, but it's okay. All speakers with us now, and we will fix a problem with Dr. Amany so soon, and we will be in track of our first, second, and third session. Then Dr. Mohamed Salah with recorded video. I'm so sorry again and apologize about this technical problem for today's session, but it's okay. We can pass it. So Dr. Bala, please start to share your screen now. You are with me, Dr. Bala? Dr. Bala? Hello? So we are waiting Dr. Bala to start sharing his screen. So Dr. Hisham, you are here with us? Dr. Hisham?
Yes, I am.
Thank you so much. So if his problem is persist with Dr.
I will
Bala,
be
do you mind to share your screen after, if you didn't fix this problem with Dr. Bala and Dr. Amany, do you mind to share your screen now?
Not really, because the others will join us from
Okay,
1:10.
no problem. It's okay. It's just
Some
okay.
will join.
No, don't mind at all. It will be fixed soon. It's okay.
Okay, thank you.
Thank you. So I will disconnect. So we will have break for 15 minutes till our next speaker will be prepared. I think today we have some. Dr. Amany is here. Okay, so I will. Okay. So I'm so sorry about this lag. We will start now. We will welcome, sorry, Dr. Amany. Dr. Amany is researcher assistant chemistry department, Faculty of Science, Moeunfia University, Egypt, and a master degree student at University of Suffolk, United Kingdom. Dr. Amany currently participating in project development of affordable kits for COVID-19 early detection, Agricultural Genetic Engineering Research Institute, Giza, Egypt. Moreover, she has participated in many other projects at GEBRI and with Heart Research Institute, Egypt. So, welcome, Dr. Amany, and we are so sorry about the experiences this connection. I will share my screen for your presentation from my side. It's okay. You see your presentation now? I don't hear you, Dr. Amany. May you unmute yourself?
Hello?
Yes, I hear you now.
Okay, thank you. Thank you. Thanks, Melam, for your efforts. Yeah, everything is going well. And I'm so honored to be with you today. Let's start.
Yes.
Thanks, Melam, for all professors here. And let's start. In this presentation, I will review and attempt to make a comprehensive list of the current COVID-19 diagnostic tools and explain why the PCR COVID-19 report is a misguided results. Next. Globally, until designing this presentation, the total confirmed cases were over 19 million cases, with the total confirmed death is near to 732K. The infection fatality rate was 3.67%. According to WHO report, and until the last month, the percentage of infected males was 53% in contrast to female proportion, which was 47% only. The infection fatality rate in Africa was 1.79% until the 6th of August, whereas the Eastern Mediterranean ratio was 2.63 only, which is nearly close to the ratio in the Western Pacific. Meanwhile, the Americans have a highly significant and a considerable ratio that reached to hit a peak of the global ratio. And I'm trying in this presentation to explain why. So, what about our fighting tools? Unfortunately, we are facing a new clinical pandemic disease, and we have no effective treatment until now or a reliable tool that can be used to cut down the exponential growth rate. At the beginning of the current crisis, the sensitivity of the PCR kits was 60, 70, and reached at the final to 90% in the sputum samples only. And the performance of the PCR kits ranged between 50 to 100%. Consequently, the economic situation got affected negatively, and the index of industrial production declined by 30% in the high-income nation, which in turn made a negative sound on the others. Next. To explain why negative PCR results is a significant sign that must be taken in our considerations, first, regarding WHO, mode of transmission is varied in its pathways, and it causes symptomatic and asymptomatic infection in some people and others in others. It causes asymptomatic infection may develop to no symptoms at all and remain an asymptomatic infection. As a figure shows, up to 50% of infected COVID-19 people aged between 25 to 64 years. Next. So, what about the common diagnostic tools to affect or to manage the pandemic disease? Clinical symptoms combined with radiology, chest CT, and laboratory. And I'm going to be focused on laboratory tests. But what are the most accurate diagnostic tools? Next. Next. Genome, SARS-2 genome encodes different types, as we know, structural and non-structural proteins. Structural proteins like spike, nucleocapsid, envelope, and membrane, which are responsible mainly for immunoinvasion, antibody neutralization, viral pathogenesis, and cell attachment. The non-structural protein, RNA ribopolymerase and proteases are responsible for viral assembly, transcription, and replication, and so on. Next, please. Regarding Professor Wang et al. At Michigan State University, they collected 6,156 genome samples across 11 countries. And they found that there are 5 clusters of COVID-19 based on highly mutations that occurred in 8 different proteins, structural and non-structural. Next, please. This study is clustered COVID-19 into 5 subtypes. The cluster number 1 is dominant and at the early of crisis. Cluster 2, which is dominant in Asian countries like Japan and Korean. Unfortunately, cluster 3, 4, and 5 are dominant in European countries. Unfortunately, United States, Canada, and Australia, unfortunately, have have all subclusters. Next, please. Okay. This study found that the highly mutation ratio occurred in 3 dominant PCR-based viral proteins: genes, nucleocapsid, envelope, RNA polymerase in the region OR, open frame reading region. And they reported that the nucleocapsid protein is the worst choice to be detected as it has a highly unique mutations or variations along the gene. Also, as we know, spike protein is the most divergent with highly mutation or highly variations among the genome. Similarly, RNA polymerase gene has also more than 200 mutations. Just only the E protein has 13 mutations. This may be responsible for the false negative results in some patients, maybe. Let's go to the next slide, please. In the second meta-analysis study has been published in WHO, done by Okayama et al., that they downloaded the more than 10,000 genome sequence from databases and across 68 countries, and they could identify that the distinct genome variations are reached to more than 5,000. Particularly, that happened in the spike protein and the open frame reading regions. Next. So on the other hand, what about the real-time commercial kit that got approved from FDA or recommended by WHO? Unfortunately, the most common kits in the market are mainly designed and focused to detect and to target N protein and the open reading region also. In addition to, next please. In addition to the high mutations that are found in the SARS-CoV-2 genome, there are some technical limitations relevant to PCR molecular-based tool, such as the long turnaround time and time consuming. Why? As it is not portable and hard to implement in the low resource setting, and also it mainly localized at the central medical laboratories. The sample collection and the transport condition may attribute to this issue. Furthermore, the nature of COVID-19 infection, the window phase, the PCR may provide a false negative results at this period in some cases, particularly asymptomatic person. And also, it relies on the kind of sample, such as the sensitivity is high in sputum sample and reach to 97% and be less in the nasal swabs. However, it's still a golden method in diagnosis or in diagnosing of positive cases and the symptomatic one. Next, please, Dr. Ola. To sum up this issue related to suspected cases, however, using the PCR are the first one, the approval kits are designed to target the most conservative regions in gene is highly mutated and they may cause several off targets. This is the first reason. The second one, about 200 of COVID-19 tested kits have no FDA approval or a WHO recommendation, and all of them have the research use only certificate, which in turn affect the quality as they have no validation. As we know in our laboratories, the reliable kits must contain internal controls, positive or negative or both, for confirmation. The third one is test time and transport condition. As we know, the lack of medical services in low regions or low resource regions may affect the transport sample, the quality of the sample. And also, finally, the nature of genome of the COVID-19 cannot be detected after 15 days of infection. So there is an urgent need to reestablish the robust kits to dedicate primers to detect the vast coronavirus genotypes to medicate the transmission and rightly diagnose. Next, please, Dr. Ola. CT provide a reliable results with a high sensitive comparing to PCR at the early phase of infection, despite it has a low specificity and low positive likelihood ratio. Please, Dr. Ola, next. Clearly, to date, there is no well-established diagnostic tool to rightly diagnose the suspected cases, as the signs and the symptoms are not entirely exclusive to COVID-19. So, how we stop spreading of COVID-19 and avoid placing others at risk? From my point of view, the serology test is an ancillary tool that may be help in this situation and may be a reliable tool also, as it depends on the antibodies against the pathogen, and the COVID-19 has flipped everything in our world. It has a capability to evoke the immune system to produce IgG and the IgM immediately at once or subsequently in the circulation. Disappeared between 2 or 3 months, but till now the longevity is unknown. Professor Long et al. Showed the antibody titer in severe and non-severe cases, which are the same, and increasing gradually in the circulation. Also, Professor Dr. Dex et al. In his meta-analysis and the large systematic review, yes, before? Yes. Yes, thank you. Reported in their meta-analysis study and systematic review, the sensitivity of antibody IgG and IgM per week. They found that at the first week, the sensitivity is less than 30%. After 2 weeks, the percentage increased to reach up to 72%. After 3 weeks, the sensitivity increased and reached up to 91%. After 1 month, the sensitivity will be 96%. Next, please. However, ELISA has been developed since 1971. It's still unused test in detecting our pandemic disease. Professor Len and Go and their colleagues presented the titer of antibodies after onset symptoms, and both antibodies are stimulated immediately after 1 week of infection, or maybe less in some cases. Next, please, doctor. A study by Kipana, which is a systematic review, it summarized the relation between the phase of infection and the best type of test in all periods. First table shows that the PCR test has a closure rule in the window phase and the consequences other period. However, it has some limitation in detecting the virus in the early stages of infection. The IgM has a powerful rule in the early phase and active infection. Meanwhile, the IgG has a capability to be a guide in the late or recurrent infection. The second table conclude. Yes, we are here. Yes, thank you, Dr. Wale. The second table concluded that the main serological tests that are validated by FDA and their sensitivity and specificity, the most of them are providing high sensitive and specific results in early infection. Next, please, doctor. So why we don't use a serological test as a reliable tool or use it alone? Because it has many drawbacks, like low sensitivity in the first week, depending on the viral activity status, and they develop false positive results in some cases. On the second angle, it provides a friendly used method and a short turnaround time, so we can implement it in high burden areas with low incomes and use various kinds of samples like blood plasma, which is considered as a non-invasive tool, and to stratify people into risk or not, or carrier cases or asymptomatic cases. Overall, we can use it for screening purposes in vital places. Next, please, doctor. Now, we need to utilize and apply a novel platforms or update the current tools to risk stratified protocols to manage the infection and the transmission. So what are the promising tools? Next, please. In my opinion, CRISPR and loop-mediated isothermal amplification techniques, both of them provide such a unique diagnostic platform because they are cost-effective, portable, minimal training, and they can be applied on low resources setting and reporting results within 1 hour. For LAMP detection, LAMP technique, it is ultra-sensitive tool with a high specificity, as it relies on 6 primers to detect 8 targets in the same reaction. Both of them reporting and enhancing the reporting quickly. And this the image at the below from Dr. Fang Zhang laboratory showing lateral flow SARS-CoV-19, which is done by CRISPR technology. Next, please. In conclusion, to improve the approach, more accurate results, and enhancing the early reporting, we need to utilize more specific COVID-19 and dedicated primers convenient to each subtypes, and along with serological test, parallel to clinical symptoms and the CT images. Thanks. Next. Next. Thank you, and I'm willing to discuss.
Thank you so much, Dr. Amany, and really amazing presentation. And I will be so sorry if we missed these great PowerPoint slides. So thanks you for, and thank God.
I'm sorry for this issue.
Yeah, no, it's okay. No, you need sorry. You can stop sharing your screen now.
Okay.
Oh, sorry. I forget that I am screen. I forget that I
have no screen, unfortunately.
I haven't screen. So sorry. But
I hope you have a one.
Nice. And you know, in the first day of our symposium, Dr. Red Herb from System University, also I was proud and I was honored to share his presentation from my side. It's okay. And my dear professor, and
yes.
So I was so honored to be his I don't know what it's called, but he's sharing his screen from mine. So you are the next.
Yes,
now.
He inspires me also. He inspires me also to do to mimic his behavior again.
Thank you, Dr. Amany, and thank you so much. Your presentation was amazing.
Thank you very much. Thank you very much.
Yes, thank you. It's my honor.
Thank you.
And today I pleased to do for you this, Dr. Amany. So don't never mind and don't say sorry, abadan. You are welcome.
Thank you.
I have a question from Dr. Subhi. So Dr. Subhi, may I ask your question, please?
Yes. Hello, Dr. Subhi. I was.
Hi, Dr. Amany. Very good, Dr.
I was
very
your
good, Dr.
students.
Amany.
Thank you. I was your students.
I am very happy.
She is your student, Dr. Subhi.
I was glad to be with you. I'm your student, Doctor.
Enjoy it.
You are my professor.
Thank you. I enjoyed with your lecture.
Thank you.
Really.
Thank you, Doctor.
Very good and lecture.
Thank you.
So
it's
I want to, because what you are said in this lecture apply firmly what I called it in the first lecture for me, because
yes.
I expected the viruses have a mutation, and
yes.
Also each day has a mutation.
Yes, highly mutated.
So this confirm exactly what you are said now. Yes. Because, and I explained why is that every day this virus will have a mutation due to horizontal gene transfer of microRNA from host, from any organisms, and so. And I see a lot of mutations. You are presented it in this your lecture. This is right 100%. So yes. This is very dangerous virus. And also the PCR, you know, the PCR give negative results now because the virus has a mutation, and this mutation will lead to the design primer for
yes.
this. It will be confirmed, and so. This is very rapid, and a big problem.
Technical limitations, yes.
This
that's
is
right.
a big problem. I mean,
yes.
This is so. I am very happy with lecturer.
Thank you.
My question, Dr. Amany, how the CRISPR-Cas9, how you use it in diagnosis? The last slide for you, CRISPR-Cas9.
Not Cas9, we will use Cas12 because Cas9 mainly used in the treatment, not in diagnostics.
Right.
Cas12
right.
and the Cas13, both of them used in diagnostic. So it depends on the lateral cleavage link between double strain or double strain of the viral
you know,
or single strain.
CRISPR-Cas9, the technology of CRISPR starts from bacteria. We are
Yes.
learning from bacteria. So
Yeah.
the bacteriophage will attack bacteria.
Yes.
Bacteria
Immune
take a
system.
bite from its DNA and put it
Yeah.
inside the genome. This is
Yeah.
inside or between two palindromic repeat and beside
Yes.
Cas9. So
Yeah.
when the bacteria, we are learning from bacteria. Bacteria learn us the CRISPR. So
Yeah.
when in a bacteriophage attack bacteria, the Cas9 with guide RNA and with the complementary space from the and
Guide.
attach with us and break down the
Clever.
virus.
Yes.
This is so I told you we are learning from animals. We are learning from
Yeah.
bacteria. We are learning from all of that.
Yes.
So I
Really,
believe
scientists.
so I believe we take some part of the viruses in a part, and when we take a sample from anybody and put it, it will be combined with it or made bearing with it and with fluorescent.
Yeah.
So this is easily and quick diagnosis.
Yes.
And
Yes.
you have a
It's
right,
right.
Dr. Amir, and I am very,
Thank
very happy
you.
with your. I am very happy.
It's my
Thank you.
pleasure. Thank you. Thank
Thank
you.
you.
You're totally
Thank
welcome,
you.
Doctor. My professor. Thank you.
Thank you so much, Dr. Amany, and amazing lecture.
Thank you, Dr. Wala.
And I wish I asked
Thank
you
you.
question about your presentation, but you are expert in this science. So, Dr. Sobhi,
No.
ask all other questions. So
I would like.
yes, thank
Yes,
you.
I would like finally to express my gratitude to my professor, Prof. Dr. Ibrahim Tantawi also. I wanted to send a deep thanks to him for providing me such an awesome opportunity like this one. Thank you.